Hello everyone, I'm Jaswinder Kaur, and in this video we will be discussing about DNA replication in case of prokaryotes, and we will be discussing about the initiation stage of DNA replication in this video. So let's start with the video. First of all, in the video I would like to discuss about the different replication models which were proposed. So there were three replication models. The very first is the conservative replication model, the second one is the dispersive replication model, and the last is the semi-conservative replication model. Now let's understand how these replication models are different from one another. So here we can see that the very first is the conservative replication model. In this we can see this is the original parental DNA, and as soon as the replication gets over we can see the two daughter strands over here. Now in case of conservative replication we see that the DNA, the parental DNA, they act as a template strand, but at the end of the replication we can see one of the daughter strand is completely similar as to the original strand, whereas the other daughter strand is totally a new synthesized daughter strand. So this is what the conservative replication model means. Next is the dispersive replication. Here what we can see, the original parental strand is there, and we can see after the replication two daughter strands are here. Now what actually happen in case of dispersive is the original parental strand does not completely or partially it get transferred into the daughter strand, but it is distributed into the daughter strand in a dispersive patch like manner. You can see small pieces of double stranded parental strand in the daughter strand. So this is how did the dispersive replication model occur inside a cell. Then is the last one, semi-conservative replication. Again you can see here that this is the parental strand, and after the replication we can see the two daughter strands. In this case also the parental strand will act as a template, but what will happen here, each parental strand will be transferred to the individual daughter strand. So you can see the one of the parental strand has been given to the one daughter strand and the other one to the other daughter strand. So this is how these three replication models are present there inside this organism. But most of the time it's been seen that the semi-conservative replication model occur in most of the prokaryotes and eukaryotic systems. So this is how the different replication models are present. Next are the prokaryotic genome. So let's get introduction about the prokaryotic genome. So this is the bacterial cell, and as you can see the bacteria have two kinds of DNA. One is the genomic DNA, another one is the plasmid DNA, which is also known as the extra chromosomal DNA. So we will be not talking about the plasmid much here. We will be more focused on the genomic DNA. So genomic DNA of a bacteria is usually circular, and it is also named as genophore. It's just another name of chromosome. Like in case of eukaryotes we say that they have chromosomes. So similarly in case of prokaryotes we name them as genophores. So their DNA is circular, and whenever the replication has to be initiated, so it always starts from a particular region of the DNA, which is also known as the origin of replication, and exactly from where the initiation actually begins. So you can see the initiation has begun from this particular area, and after some times this replication is being initiated on a smooth basis, and you can see the daughter strands are being synthesized. So this is the new DNA, the red one, and the old DNA. So this is how the replication actually is being initiated in the bacterial genome. Now the DNA replication is always bidirectional. This has to be very clear before understanding about the replication process. So DNA replication, either it is happening in case of bacteria or a eukaryotic cell, it will always occur in a bidirectional fashion. So always there will be two replication forks formation. So there is nothing which is known as unidirectional replication. The replication will be bidirectional. Let us begin now with the replication part. So replication occur in three stages usually, and which is the initiation, elongation, and termination. So let us understand with this diagram how the initiation, elongation, and termination actually are occurring, in what manner they are occurring inside a cell. So this very first picture is the stage 1 of the replication, which is the initiation. Here we can see this is the origin of replication, and the initiation, how does it occur? So at the time of replication, so what happened, origin of replication is being recognized by various proteins and enzymes. So all the recruitment of all these proteins are being done, and this makes an initiation complex. So this is how the initiation stage of the replication occur. Next are the second stages of the replication, which is elongation. Here we can see that the initiation part is completed. The complex was initiated to the elongation part, and where we can see the formation of a bubble-like structure, and two replication fork are seen on the either side of the region of replication. The daughter strands are being synthesized with the help of DNA polymerases, the major enzymes which play a role in the replication process. So here we can see the elongation part. So in the third picture we can see the 50 to like approximately 60% of the DNA is being replicated, and just this part is being left. Last stage of the replication is the termination stage. Here what will happen, the replication has to be stopped now. It cannot be continued for the longer time, because now for now the purpose of cell is to duplicate the DNA so that they can produce two daughter cells. So for that reason, the termination of replication is very important, and it happens around a ter region which is present in the genophore of the bacterial cell. So this is how finally after the termination the two daughter strands will be separated out. So this is what actually happened on a broader picture, if we can say. So this is what happened in the replication. Now let us begin with the understanding of initiation in a more broader picture. So here is the initiation. We will take an example of E. coli cell to understand more about the initiation portion. So see, one thing before starting is important, which is, see, if there is one bacteria, so obviously it will have one genomic DNA, and one genomic DNA will only consist of one ORI. So this is very important to understand. There will be only single ORI present in a genophore. So this is actually the ORI region, which is been shown over here. Now this ORI region is approximately 245 base pair long. So this is the length of this ORI region. Now this 245 base pair DNA will have different segments, or I can say motif present on the DNA, which will be recognized by the DNA A protein, and the initiation of the replication will start by this manner. Now to understand more about this, let's have a picture. Yeah, this picture. In this picture we will be having a proper understanding about the initiation path. So again, you can see over here, this is the ORI region, which is approximately 245 base pair long, and I was saying there are certain motifs which are present. So these motifs are 9 mers and 13 mers. Now what are these 9 mers and 13 mers? Let's understand. So 9 mer is nothing but a 9 base pair long sequence which are being repeated on the origin of replication for approximately 4 to 5 times. Similarly, 13 mer is a sequence which is 13 base pair long and being repeated on the ORI region for like 2 to 3 times. So these are the two motifs which are present. Now what happen is, initiator protein, that is the DNA A protein, will come and bind onto the 9 mer region. You can see these are binding onto the 9 mer region, and as soon as they are binding, they are containing, or they are like starting a negative supercoil stage, due to which what will happen, the 13 mer sequence which were present just upstream of these 9 mers will start a stress. So a stress will actually start in that region, and due to that stress or tension, I can say, the unwinding will occur. What will happen? The unwinding will occur, and due to this unwinding, the two strands of the DNA are now being exposed. As they are exposed now, what will happen? The DNA C, the helicase loader, will load the DNA B, the helicase, and what will it do? It will place the DNA helicase onto the unwounded or exposed DNA strand structure. As soon as the helicase are bound to the DNA strand, what will happen? They will activate the DNA G protein, or we can say the primase. Now what these primase will do, they will start the addition of RNA primer onto the strands, the leading and the lagging strand. So this is how actually the initiation will occur, and you will also see as the DNA strands are being exposed, the unwinding of the DNA strands are happening. The SSB proteins are going to bind at those single-stranded DNA. Like how, if I draw something like this, so this is the bubble formation, or I can say the replication fork formation. See, and these are the helices, they are moving to this direction, and these unexposed part or the single stranded DNA, they will be covered with the SSB proteins in such a manner so that there is no re-annealing happen and the replication process goes smoothly. So this is how the actual initiation takes place inside a bacterial cell. Now to understand on a broader picture, how does that DNA looks like when the initiation complex is being formed. So it looks like this, something like this. We can see the leading strand and the lagging strand are being formed, the whole replication fork structure, and also we can see the primase and the helicase are present over here. So you can see the helicase is moving in this direction and unwinding is being done by it. Now how do the helicase do the unwinding? Simply by breaking the hydrogen bonds between the nitrogenous bases. So this is how the two strands of the DNA are being separated. Now as the separation is being catalyzed by helicase, the primase will add on the RNA primer to the leading strand and the lagging strand simultaneously. Okay, next the topoisomerase is present upstream of the helicase. So what is the function? The helicase is going to relieve all the positive supercoil which is being created by the function of helicase. Now helicase is continuously unwinding the DNA, and due to that unwinding, the rest of the DNA will face some kind of positive supercoil or a kind of stress, due to which it might disrupt the function of helicase. Helicase may not be able to function accurately, and it may degrade from its original structure, and this will ultimately lead to the replication halt, which cannot be afforded by the cell. So what cell does is it plays a topoisomerase so that all the positive supercoiling which is being created with the functioning of helicase could be relieved, and proper smooth process of the replication is being performed. So this is how here the replication is being initiated. So that's all for this video. Thank you so much.